Hello everyone and welcome to the screencast on capillary exchange physiology. In this screencast we're going to focus primarily on the exchange of fluids between the blood and the body tissues that occurs at the level of the capillaries. So let's go ahead and get started. Here's a diagram that we're going to use to represent a capillary in a capillary bed. This would be a single capillary from the capillary bed. And of course, whenever we take a look at this capillary, we note that capillaries are made up of walls that contain only a single tunic. And that single tunic is the tunica intima or the endothelium. The tunica intima or endothelium is made up of simple squamous epithelium. And so as we take a look at our diagram, these are going to represent the squamous cells that make up the capillary wall. Note that the squamous cells have little gaps between them. That was intentional because whenever we do take a look at the capillary beds, we do note that the squamous cells do have little gaps between them in the actual capillaries. And this is going to be very important for our understanding of capillary exchange. All right, on the left side of your screen here, we're going to represent the arterial side of the capillary, and on the right side is going to be the venous side of the capillary. So let's add in some tissue cells that represent the tissue that the capillary bed is allowing blood to flow through. And inside the capillary, let's add some blood cells and some proteins. Now, there are many different types of proteins. I want to focus on them for a minute. There are many different types of proteins that we find suspended in the plasma of the blood. But we're going to focus on one type of protein, albumins, because they contribute the most and are very important in understanding capillary exchange physiology. All right, so the blood's going to be moving through the capillary now from left to right, obviously from the arterial to the venous side. Let's take a look at two factors that actually affect capillary exchange. The first factor is blood pressure. And we note that the blood pressure on the arterial side of the capillary is higher than it is on the venous side of the capillary. That's because as we move across the capillary from arterial to venous side, we're getting further and further away from the heart. The heartbeat provides the force that pushes the blood through the capillary. The further you are away from the heart, the lower the blood pressure is going to be because you're further away from the force that's pushing the blood through. All right, another factor that's going to affect capillary exchange is osmotic pressure. Osmotic pressure inside the capillary is going to be lower on the arterial side, but on the venous side, the osmotic pressure is going to be the opposite of the blood pressure. It is going to be higher. So I think we can understand why the blood pressure changes from one side of the bed to the other. But the osmotic pressure is a little bit of a different story and requires a little more explanation and some background to understand why it changes. So let's take a look at that background. And of course, osmotic sounds like osmosis. As a matter of fact, osmotic means pertaining to osmosis. TIC is a suffix that means pertaining to, and osmo means osmosis. So anytime you consider osmosis, and osmosis, by the way, is defined as the diffusion of water across a membrane, or the movement of water from where it's in high concentration to where it's in low concentration as it crosses a membrane. But anytime you talk about osmosis, there are three important terms to consider. The first of these terms is hypertonic. Hypertonic is a term that refers to a solution when compared to another solution that has a higher solute concentration and therefore a lower concentration of water. The next term is hypotonic. Hypotonic is a term that we use when comparing solutions to mean a solution that has a lower solute concentration and therefore a higher concentration of water when compared to the other solution. And the final term is isotonic. Isotonic is a term that means the solution has equal concentrations of solute and water when compared to another solution. Okay, so let's show you what we mean by this graphically. And here we have two beakers. And we're going to fill each of the beakers with 500 milliliters of distilled water, let's say. And we'll call the beaker on the left Beaker A, and we'll call the beaker on the right Beaker B. So now let's add some solute to Beaker A. Maybe we'll take some salt and mix the salt in with the distilled water and dissolve the salt in the distilled water. So here's the amount of solute that we added to Beaker A now. And let's do the same thing in Beaker B, but we're going to add a little bit less salt. And so here's the amount of solute in Beaker B. So which solution, the solution in Beaker A or B, would be considered hypertonic? Remember, hypertonic means has a higher concentration of solute and a lower concentration of water when compared to another solution. Well, I think you can see that Beaker A has the hypertonic solution. It has a higher concentration of solute than Beaker B. So if we take a look at Beaker B, we would say that Beaker B is a hypotonic solution. So we're comparing these two solutions to one another. Okay, now here are our two beakers again, and Beaker A is hypertonic and Beaker B is hypotonic. Let's take these two beakers and push them together, and if we could, let's suppose we could, cut the glass between the two beakers, and let's add a semi-permeable membrane between the two beakers. Remember, a semi-permeable membrane is one that will allow some substances to pass through while keeping others out. So now the question becomes, if we put this semi-permeable membrane between the two beakers, in what direction would the water move? Well, remember, water is always going to move into the hypertonic solution. So as we take a look at these two, water is going to move from Beaker A into, or excuse me, from Beaker B into Beaker A. Why does that happen? How do we know that? Well, where we take a look at it, if we look at Beaker A, we can see the amount of solute in there. And remember, solute is matter. Matter has mass and takes up space. So the solute and the water molecules cannot take up the same space. The more solute you have, the less room there is for water molecules. So in Beaker A, there has to be less water molecules than in Beaker B, because Beaker B has less solute, so there's more room for water molecules. And given the fact that these are both 500 milliliters of water, then there has to be more water molecules in Beaker B. So the water is going to move from where it's in high amount to where it's in low amount, from Beaker B to Beaker A. Water always moves into the hypertonic solution for this reason. And so there we can see that what would happen is the amount of water in Beaker B would go down while the amount of water in Beaker A would go up. In other words, Beaker A would become more watery or aqueous, and Beaker B, as a result of the loss of water, would become less watery or more concentrated. All right, how does this apply then to fluid exchange at the level of the capillaries? Well, what we're going to see is as we move from the arterial side to the venous side, the blood pressure is going to be higher. And when the blood pressure is higher, that's going to cause water to be pushed out of the capillary bed between the gaps of the simple squamous cells and into the interstitial fluid. So that what's going to happen is as the water squirts out, so to say, into the tissues, the interstitial fluid is going to become more watery or more less hypertonic and more hypotonic. while the blood plasma is going to start to become more and more hypertonic as we move again from the arterial side to the venous side. So we can see with our beakers down here that the beaker on the left, which represents the interstitial fluid, becomes more watery, while the beaker on the right, which represents the blood plasma, becomes less watery, more concentrated, and becomes more and more hypertonic. Another way to look at this is graphically. So let's draw a little graph on here, and let's represent what's happening on the arterial side as we move from the arterial side to the venous side to the blood pressure and the osmotic pressure. So we'll plot on our graph blood pressure and osmotic pressure. And of course, as we move from the arterial side, the blood pressure is going to drop because we're moving further and further away from the heart. But what about the osmotic pressure? And we're primarily looking at osmotic pressure inside the blood vessel here. As we move from the arterial side to the venous side, what's happening? The blood plasma is losing water as we move from the arterial side to the venous side. And as a result, the blood plasma, as we move from the arterial side to the venous side, is becoming more and more hypertonic. So if we draw a line here, we can see there's a point where they intersect. And after they intersect, the blood pressure now has dropped so low that there's not enough force to push the water out of the blood vessel, while the osmotic pressure now is flipped and is higher, and that osmotic pressure causes the water to flow back into the capillary bed on the venous side. Remember, water always moves into the hypertonic solution. Okay, so back to our diagram here. We take a look at the diagram. We ask ourselves the question of the water that squirts out on the arterial side, how much of that water is recovered on the venous side? And the answer to that question is about 90% of the water is recovered. Not all of it, and about 10% is not going to be recovered from the tissues. Well, that creates a bit of a problem, right? If we don't recover all the water that squirts out on one side, on the arterial side, if we don't recover all that water on the venous side, then what's going to happen is the tissues are going to begin to inflate with water, and edema is going to happen, or the tissues are going to become swollen. So the question is, what happens to that other 10%? We need to get that other 10% returned back to the blood as well. And of course, here's our graph that shows us why only about 90% is recovered. If we take a look here on the left side of the graph, we can see that our line where the two lines intersect one another is moved to the right of the graph. So there's a longer period of time where the water is being squirted out, and a less period of time or distance that the water is actually being recovered. So not all the water gets recovered on the venous side. So what happens to that extra water? How does it get recovered? The answer to that question is the lymph capillaries. These lymph capillaries that we find spread in and among the capillary beds are going to recover that excess water and help to keep the tissues with the right amount of waters in them and keep edema from occurring.